As we move forward, we now begin the next session of the day. We are pleased to have Dr. Rupesh Deshmukh with us today. Dr. Rupesh Deshmukh is a professor and head of the Department of Biotechnology at Central University of Haryana, Mahendragarh. He leads a dynamic research group that has made significant contributions to our understanding of the aquaporine transport system in plants. Dr. Deshmukh's group is also at the forefront of the genome editing research, tackling several challenging projects aimed at translating genomic insights into practical applications for crop improvement. With over 200 research articles published in reputed peer-reviewed journals, his scholarly output reflects both depth and breadth in plant biotechnology. His article got cited over 11,000 with H index 57. He serves as an associate editor for several prestigious journals, including Journal of Advanced Research, the Plant Genome, and Journal of Plant Growth Regulation. His outstanding contributions have been recognized through numerous accolades, such as the Ramalingaswamy Fellowship, the NASI Scopus Young Scientist Award. He is a fellow of the Maharashtra Academy of Sciences and currently serves as secretary of the Society for Plant and Agricultural Sciences. His name has been consistently featured among the top 1% of scientists in the Clarivate list, one of fewer than 10 from India. It's now my pleasure to invite him on the stage to deliver the next lecture of the day. Dr. Deshmukh will be sharing his insights on biofortification, a tool for improving food security. We are truly looking forward to learning from his experience and expertise. Over to you, sir.
कोई ठीक है, यहाँ से कर दो, लेजर चलेगी तो भी ठीक है। Thank you very much for such a wonderful introduction. Already you have attended several classes, so it is pretty much clear for you to how nutrition is important for everyone's life. So today we will be talking about the biofortification and more particularly the new approaches which we are being used for the biofortification in different crops. So fortification we use at a very large scale in industries to make food more nutritious and to tackle with the nutrition deficiency mostly which is known as a hidden hunger. And biofortification is a relatively more safer and sustainable approach where we can directly modify our food in a way that which will provide us a required nutrition and which will help us to tackle with the hidden hunger. So you are already, I guess, familiar with this situation that the global scenario of the hidden hunger is like 1% out of 3 is suffered with the hidden hunger, and then these deficiencies are mostly with the vitamins and minerals. People are getting required calories with the different foods, but most of the time they are suffering with the deficiency with any specific minerals or vitamins, and that is the like an emerging problem throughout the world, whether it is developed country or whether it is developing country like us. And these problems are more prominent in case of small children's. Here you can see that there are the symptoms like impaired brain development in children or stunting growth of the children's, and more than 2 billion people worldwide suffer from the hidden hunger, and more than 1 million children under 5 dies every year because of the vitamin A and zinc deficiency. So this is a very huge issue worldwide. Even if we are, the world is getting better and better every day in terms of providing sufficient calories to the growing population, but till we are lacking with the nutrition quality of the food. So these are the major approaches which are being used for the improvement of the nutritional quality of the food. So here you can see food to food fortification. Oh, mic है वो वाला. Hand mic है. इस माइक में बहुत क्लोज रहना पड़ता है. हाँ फिर चेंज करने में दिक्कत हो जाएगी. अच्छा अच्छा ठीक है. Okay, so these are the major approaches which we use to fortify the food. So these include like a food to food fortification. Here basically we use our traditional knowledge, which is already where we in India, more specifically in India, we have several different combination of food, and that they supplement each other to provide us a complete nutrition. Like we mostly consume like dal chawal. So one is rich in a carbohydrate source, second is rich in a protein source, and we also use the different spices to make it more tasty for us, but that also provide different nutritions. That is a better source for minerals, that is a better source for vitamins. And some example, like we also eat multigrains that include cereals as well as pulses. So they supplement each other to make it the uh, bread more nutritious. Second approach is like a conventional fortification, where we use fortification at the industry level. Like food industries, they use different fortifications, like they use calcium to supplement in the milk, they use iron to supplement in different type of flours. So this happens at the industrial level. And then the dietary diversification is also a good approach to tackle with the hidden hunger. Like if we include a diverse food in our daily diet, that will also take care of our nutritional requirement. And then the approach like a biofortification, which is the main focus of today's talk, that is one of the sustainable approach, and that is also a safer approach. For example, when we do fortification at industrial level, the amount of required minerals or vitamins, those are not very precisely monitored, or there could be mistakes or errors happens at the industrial level which will affect a large population. Second important thing, when we do fortification at the industrial level, there is always concern about the bioavailability. Like when we take such a nutrition of vitamins or minerals through the fortification and industrial products, we that is not guaranteed that that will provide you a sufficient nutrition, because the bioavailability of such products are not very high, and that this concern we can easily rectify with the approach like a biofortification, where we basically develop new varieties which are more nutritious. We can feed our dairy animals with the better minerals or vitamins, so that the dairy product will get a better, or even we can have this fortification at the different level, but through a mean of biological agents. Like if we use fertilizers in crops, the crop will get a better amount of zinc or iron, and then that will enter into the food chain, and that iron or zinc has a better reliability compared to the fortification at industrial level. Oh, अच्छा, ठीक है. Okay, so with here you can see what approaches we have at a biofortification level. So we can use genetic engineering. In genetic engineering also there are several approaches. Like when we are talking about genetic engineering, that provide us options like a cisgenetic, that provide option like a transgenetics. That will be like if we can use a specific technique like RNAi, or to shut down any specific gene which will improve the nutrition quality. We can take a gene from any different organism and express in a plant, that is transgenic approach, so that we can improve the nutrition quality. And now recently recent improvement or recent advancement in biotechnology, that is the genome editing. We can use genome editing, which will be a transgene free, but that will enhance, that will make the food crop as we desire. That will make desirable changes in the food crop so that we can easily improve the nutrition quality. And then some approaches, those are sustainable and very easier to adopt. Like we can use plant growth promoting bacteria, or maybe zinc solubilizing bacteria, iron solubilizing bacteria, our organic farming system, so that they can improve the uptake of these minerals and we will get a better nutritious food from that. So next slide. Yes. Here you can see that, oh, I think it is very difficult to read, but I will explain you. There are several minerals which are important for us as a nutrition, but those are equally important for cropland. If we use those minerals in a fertilizer, they will also benefit plant growth. Like if we added silicon fertilizer to crop, that will improve resistance against different diseases. That will also help plant to improve resilience against abiotic stress like a drought, salinity, flooding, those things. So it is like a double benefit. If we use such minerals as a fertilizer, that will also improve our crop, and then this will also produce a good nutritious food. So that's why it is very important to tackle to at the cropping level in agriculture. So here I have highlighted the silica because we know that the different minerals like zinc, iron, boron, those are very important, and we have already talked about it. You can find it in your textbooks also, that highlighted how zinc, iron, boron, these are the very minerals. But silicon is relatively new element. We classify silicon as a quasi-essential element. Basically, in terms of the plant growth, we categorize minerals into two categories. One is essential minerals and another is non-essential. So to make these categories, one simple trick we use, like whatever mineral is important to complete the life cycle of the plant, that is we categorize as a essential element. That means if we remove that element from the plant nutrition, plant growth media, plant cannot complete the entire life cycle. That's why that those elements are essential element. And non-essential element means if we don't use those elements into plant growth media, plant can survive and complete the life cycle. So silicon is, we keep silicon in between those two categories, like a quasi-essential. Because plant can, most of the plant species, they can complete the life cycle without silicon. But if we provide silicon, plant grows better. They have a better resilience under stress condition. That's why it is cost essential. I am highlighting silicon little bit more because my group work with the silicon uptake in plant, and silicon is equally benefited for the human health also, because there are several tonics available in the market which are silicon rich, and those are referred for the bone health. If in an old age bone get more fragile, there are more chances of getting it fractured, and for that to improve the bone health, doctors prescribes this silicon based tonic. So here in one photo silica, that is the tonic for the bone health. Okay, next slide. So biofortification, we can these approaches we can classify further as agronomical approaches and genetic approaches. So in case of agronomical approaches, we can use plant growth promoting microorganisms. As I mentioned, that we can use a zinc solubilizing bacteria, phosphorus solubilizing bacteria, we can use iron solubilizing bacteria, so that that will make this mineral available for the plant, and then it will enter into the food chain. Okay. Second, we can select good variety. Like if we screen different varieties of rice or wheat, we can select any such variety which has a more uptake of this mineral elements. So that we can do. That is also categorized as agronomical practices. And then we can increase the doses in terms of fertilizer or in terms of the feed for the dairy animal, so that we can increase these mineral elements in the plant or dairy products. Second category is a genetic approach. So One first category is agronomical approach, where we use fertilizers, microbes to just improve the mineral content uptake in a plant. And second is the approach is a genetic approach. So in genetic approach, we try to improve our crop at genetic level, because once we make any genetic improvement in a crop, that is sustainable. That is one time invest, and then we can harvest that product like several generations. Once you make any genetic improvement in a variety, that variety will give seed next for the next generation, and then we can harvest seed in the subsequent generation, so that it will be for a long time. We can harvest that product for the several generations. That's why it is more sustainable. So in case of genetic improvement, we can use plant breeding approaches, like which we already using. Like we first select any variety which is not so high yielding but has a better gene uptake. Then we cross that variety with our high yielding variety, so that we can transfer that gene uptake trait from that variety to our high yielding variety. Now in that way we can develop a new variety by breeding, so that the new variety will be high yielding as well as that will have a higher gene content. So that is a breeding approach. And in breeding, we can use some marker assisted breeding or omics assisted breeding, where we use different advanced omics tools to just speed up the breeding process. But basically, we are exploring here whatever naturally present, the allele which is present in a one variety, which help that to uptake a more gene. We are using the same gene, just we are transferring that same gene to the another variety using a natural process. That is like we make a cross pollination and then crossing, and then we follow the normal breeding process. That is the conventional approach, right? And second approach is a genetic engineering. We can make the genetic changes using advanced biotechnological tool, and then that improved variety we can use for the better nutrition. So in genetic engineering, uh, we use a transgenic approach, where we can take a gene from any other species, and then we make a transgenic, which will have the new trait completely new from borrowed from the different species. So this is not the like a conventional thing, or this is not a natural process. This cannot happen naturally, right? So this is like a completely man-made thing, which we develop for our purpose. And already worldwide several transgenics are being grown. We will discuss this in detail. So whether it is really required to make a transgenic or not, what are the alternative approaches? Like for transgenic, we have one recent alternative approach, that is genome editing. So in case of genome editing, we don't use any transgene. We just change few nucleotide in our crop with genome editing tools, so that they will be a better crop. Like we can make a desirable genes. That could be for the better nutrition, that could be for the better resistance against drought or resistance against different diseases. Okay, next one. So these are the major biofortified crop varieties in India. So basically, these are the three main crop where we have made significant progress. That is the pearl millet, where we have several varieties for higher iron content in pearl millet. Second is the zinc rice and zinc wheat. So these are the three crops where we made a significant progress. And this slide is from the Harvest Plus. So Harvest Plus is such a program where the different government organization as well as a private partner, they come together, and they are uh, trying to employ efforts to improve the crop varieties for the biofortification purpose. So Harvest Plus is basically focusing on the biofortification, and their major area of work is in South Asia, which include a largest population of the world. That includes India, Pakistan, Bangladesh, Philippines, Vietnam. These are the countries where this Harvest Plus works. Okay, next slide. This is very interesting. This is biofortification priority index that is created by Harvest Plus, and this shows that which country has required more priority on this biofortification. So at the top, this is particularly for the rice. So at the top, that is Bangladesh. because Bangladesh is in Bangladesh their major diet is rice, right? Entire country with almost 100% population eat rice daily. That's why their biofortification priority index is very high. And India is also in the high priority countries. For India also, biofortification in rice is very important because in India, almost two-thirds people eat rice daily. And the coastal population, like a highly populated state like Bangladesh, West Bengal, Kerala, Tamil Nadu, Bihar, those are the more rice consuming states. So it is very important for India as well to improve the rice. Next slide. Okay, so these are the major biofortified rice varieties in India. So in this table you can see DRR Dhan 45. So that has parentage IR 7307 crossed with IR 77080. So these are the parentage of that variety, and that is having high gene content. So all these varieties, they are developed by a breeding approach. Like we first selected one variety which has a higher zinc uptake and cross that variety with the elite genetic background, the high yielding variety which farmers are already cultivating. And then this new trait was incorporated into the high yielding variety to have a variety with a high yield, high production as well as a higher zinc content. Right. So these are such varieties. So there are many, but these are the significant varieties which has a significant cropping area. Next slide. Okay. So this is just to give you an idea how this breeding works, right? So first we identify a donor variety. That means whatever variety which has a higher zinc content in their seed, and then from donor variety we cross that variety to a recipient variety, which is high yielding variety. And after that, it takes several years to just fix that trait to make a homozygous line so that that can be released. If it is open pollinated, self pollinated crop, then we need to make it homozygous. So that also takes a couple of years, and then it will enter to the different trial programs. Like first, that will go to the preliminary yield trial, then that will go to the advanced yield trial, and then it will go to the national level trials where it need to bid to the different check variety. Then only we can release our variety. So it and taking process. Usually it takes 8 to 10. Like here, this is the different steps. So it will take 8 to 10 years to get a variety. So there are two approaches. This is first approach where we cross one variety with another one. And then we take every generation, we advance it further or make it homozygous and then evaluate at different stages. And finally, at the 12 to 13 year, we can release that variety. Another approach we have that is induced mutagenesis, right? One approach which is using already available genetic resources. Like first find out one variety which has a better trait, and then cross that with the highly variety. This is one approach. Second approach is a mutation breeding. In mutation breeding, we use a induced mutagenesis. So we take a, for example, in rice we take a seed, rice seed, and then we treat that seed with a chemical mutagen like EMS that create a point mutations, or we can send that seed to the facilities like a bark where we can radiate that seed with the gamma radiation. And with the mutagen that will randomly create a mutations, and from that randomly induced mutations, we need to identify a line which has a better trait, like for example, better gene coverage. That means at least we need to have 30-40,000 such mutant lines, and then this 40,000 mutants we need to screen for the gene coverage because it is a random process. We don't know whether that 40,000 mutant, whether any of that has a higher zinc or not. But we need to screen those 40,000 and then first identify if there is any such mutation and then use that mutant to develop a variety. So it is also time taking and it is completely based on the chance. Right. So both the processes are very time taking. There are several different approaches, but these are the major two approaches in the breeding. One is a breeding with the biparental population, or second is a induced mutagenesis. So second, next slide. Okay, that's why people started thinking about the genetic engineering. Second thing is here very important. With the conventional breeding approach, we can explore only those traits which are naturally exist. If any gene is already present in a natural germplasm, then only we can have that. Otherwise, we cannot use conventional breeding or mutagenesis to get completely new trait. But genetic engineering provides a flexibility which is not naturally present in the genome of rice or wheat or any crop. We can bring any new trait from any other species that we can do with the genetic engineering. Next slide. So the process of genetic engineering is very simple. Or I think most of the you are already at master level, so you might have already known that the process. So first we clone the gene uh, from any organism in a vectors like a plasmid vector, which we use for the transformation, and then we transform that gene to the our plant. We first clone the target gene, and then we transform that to the crop variety. So there are two major methods. One is the agrobacterial mediated transformation, another is a gene gun, right? So we can use either of the methods. Some crops are very easier for the transformation with the agrobacterial mediated transformation. And then we grow the transgenic plant, and then we evaluate that transgenic plant whether that has the desirable character which we expected or not. And then if that is giving a significant improvement in our desirable character, we try to release that for the commercial release for the cultivation in different areas. Okay, next. Okay, so these are some examples which are already commercially released, like a flavor saver tomato, 1990s, that was released as a first transgenic food crop. And for the flavor saver tomato, RNA technology was used. And then this PPO gene is also used in a potato or mushroom or apple that reduce if we reduce this PPO, that will reduce the browning in an apple or in mushroom. So thereafter, now the new era started with the genome editing, and several products are already in the market with genome editing technology. So in a subsequent slide, we can see some of these products which are created with the genome editing. So next slide. Okay, so this is just a difference like transgenic and cisgenic. We call transgenic because whenever we take a gene from any other species and transfer to the different species, that is transgenic. But sometimes we also need to take a gene from the same species and just overexpress it. So that is cisgenic. So at a commercial level, there are several transgenics, but at this point, there is no cisgenic in a commercial market. So that is at an experimental level. So transgenic is more common compared to cisgenic, because in cisgenic like RNAi, where we are using the same gene sequence to make it knock down, but we also need some selection markers with it. That's why it is also called as a transgenic. It cannot be considered as a cisgenic. Okay. Next slide. Okay. So here just a comparison of different approaches. In conventional breeding, we use this is high yielding elite variety crossed with the donor one. And then after several generations, we will get a high yielding variety with this desired trait. Second approach is mutagenesis, where we use mutagens like gamma radiation or EMS, and then from the thousands of mutant, we select a desired. Then this is the tissue culture approach like protoplast fusion, where we can make a hybrids different. We can use a related species to make a hybrid. And this is genetic modification, genetic engineering approaches, where we can use a transgene, like take a bacteria from take a gene from bacteria and then overexpress in our high yielding variety so that we can get a transgenic. Then cisgenic, this is the same variety but same species but a different variety. Take a gene from different variety in the same species and then make it express in our elite genetic background. So this is the transgenic approach, and then the new approach is genome editing, where we are not taking anything from any other variety or any other species. We are just making the same nucleotide change in our elite genetic background so that we can have the same desired trait in elite plants. Is it clear up to now? If you have any question, you can interrupt me and ask. ठीक चल रहा है? चलो ठीक है, next. Okay, this is the history of transgenics. So it started long back. Like in 1946, that conjugation was proved that one bacteria conjugate with another and transfer the genetic material. It started long before this, like with a Griffith experiment with the pneumonia bacteria. And then in 1954, that Watson and Crick model and everything become well accepted. And then the first genetic engineered product, like in 1973 and 1983, the first GM plants was developed, that is in tomato. And then in China, 1991, commercial GM was released. So any guess which commercial crop was released in 1990 in China? Transgenic crop. Guess, नाम बता सकते हो कुछ कुछ तो? Golden rice, okay. BT cotton or BT maize, BT brinjal or anything else? Beans, okay. Beans. Okay, that was the tobacco, because first genetically engineered product was also tobacco, and then China released tobacco as a genetic engineered first transgenic crop. That was the tobacco, and then in 1994, FDA approved first food crop, that is tomato flavor saver. That was the first transgenic food crop approved by FDA. Next slide. And then the first 1996 first genetically modified soybean was introduced in United States as a commercial crop. Then BT cotton was approved in United States in 1996. 2002, that BT cotton reached India. India approved BT cotton's cultivation in 2002. So you can imagine that already we passed almost 3 decades with the transgenic crops. And the recent, we can say, like a DMH, that is Dhara mustard hybrid. BT brinjal was also approved by the Indian government, but later on that was on the hold because of some concerns. And Dhara mustard hybrid, that is very recent, like that was approved by the Ministry of Environment and Forestry, but because of the concern about its yield over the checks, and also concern about the use of the herbicide, that is on hold, because Dhara mustard hybrid was first tried to release for its ability to make a hybrid crop, because mustard is a self-pollinated crop, and if you wanted to grow a hybrid crop, then we need male sterility in their parent. So Barney's bar system was used. Barney's bar, those are the two genes. Barney's create a male sterility, and bar star restored it. So Barney's bar system was used to make this Dhara mustard hybrid. And when the yield trial was conducted in Punjab University, Punjab Agriculture University, Ludhiana, and IRI at New Delhi, significant increase in yield production was observed, but that was compared to the pure line varieties, not with the present hybrid. That was one concern. And second concern, then this repurposing also tried, that this crop has a one selection marker, that is herbicide tolerance. So that was also tried, but that was not very convincing. So it is on the hold. So now my question is that how many transgenic crops are being grown in India today? कौन कौन सा पता है? Hmm? At present, which transgenic crops we are using? BT cotton, anything else? और दूसरा कोई? BT brinjal और बस golden rice और दूसरा कुछ? Okay, so today in India we have only one crop, that is BT cotton. We don't have golden rice, or we don't have BT brinjal, or we don't have the Barnett bar mustard. At present in India, only we have only one transgenic crop, that is BT cotton. And in India, more than 1,000 laboratories, they are working with the transgenic technology. And that too from the last 3 decades. So you can imagine 1,000 labs are working on transgenic from last 3 decades, and we have only one transgenic crop, that is BT cotton. And that is also not from the Indian lab, that is from the Monsanto, right? So why this is happening? Any guess? नहीं नहीं आप लोग नहीं बोल रहे, आप लोग ऑलरेडी क्लास कर चुके हो नहीं? Regulatory concerns for the GMO release. That is the main issue. So actually what happened whenever we release any transgenic crop, it is very easy to release it. But if anything goes wrong, to take it back is very difficult. Once the the seed is released in environment, that will flow with the floods, that will be contaminated with the forest or different uncultivated land also, and taking each and every seed back is a very difficult. One example is DTT. When scientists discovered DTT, he got a Nobel Prize, right? And after within a 1 decade, government banned the DTT because that realized that because of the DTT, the breast cancer incidence was frequently occurring. That caused of breast cancer. That's why it took back. But earlier, that discovery awarded with the Nobel Prize. So this thing happened. That's why it is. We need to be very cautious when we release a transgenic. Therefore, our regulations are very tight, and we just allowed the BT cotton to grow in India. That is the transgenic one. Okay, and that also helped a scientific community to come up with the new tools. Since the transgenic is not easy to commercially grow, the scientific community comes with the new technology, that is genome editing, which will produce a new plant but completely transgenic free. There will be that will be genetically modified but free from the plant gene. There will be desired modification we can do in our genome anywhere, and there is no need to have anything from the any other species or the same species. Okay. So biofortified rice using genetic engineering. When we talk about these two things are just I wanted to remind you again. These are the same slide we already seen. This is the method how we can develop the transgenic, right? We can use different transformation method and take gene from one species and express it in another species. This is how we develop the transgenic. And why? Because this is the biofortification priority index. World's half of the population lives in this country, and there is a very high level of malnutrition, very high level of hunger. That's why it is very important. So now this is very clear to us how we can do this and why need to do this, right? So second slide. Okay, so now this is the first biofortified significant product, that is the golden rice, right? And this was developed by the uh, Portus Ingolicus, and he when he developed this golden rice at International Rice Research Institute Philippines, and that was now released in 2 countries, Philippines and Bangladesh, for the commercial cultivation. It is released in Bangladesh and Philippines, where farmers are growing this in a field. Okay, and in India it is still under hold because of the regulatory norms. So this is the normal rice. When we make this having provitamin A enriched, that become a golden in color. That's why it is known as a golden rice. Next slide. Okay. So this here you can see this is the prevalence of vitamin A deficiency. So India, Bangladesh, and this is Korea, Vietnam, Sri Lanka, Pakistan. These are the countries which are more suffered with the vitamin A deficiency. Uh, like millions of children, they are affected with this deficiency of vitamin A, and that is the reason why this golden rice is very important. So next slide. Okay. So for to develop golden rice, 2 genes were taken from daffodil, 1 gene of taken from the Irvinia, and then those 3 genes, 2 from daffodil and 1 from Irvinia bacteria, they expressed in a rice grain with the endosperm specific promoter, and so that can produce this provitamin A in the rice, and that then the rice become a provitamin A rich. So second slide. Okay, so there are several generation of golden rice. So in first generation, you can see here, this is a wild type rice, white, clear. Then first generation golden rice that is having a little bit golden color. And then second generation is a more yellow in color. So in first generation, three genes were used, two from the daffodil, one from Irvinia. And then second generation, two genes were from the corn and one from Irvinia. And there is third generation also. So with every generation, the content, provitamin E content is improved. Next, okay. So these are the gene phytoin synthesis, and the second one is phytoin desaturate. So these are from the maize, and then those transform to the rice so that this provitamin E increased. Second, okay, this is the pathway, that is grenyl diphosphate pathway, and these genes work here. Like rice lack these four genes in this pathway, which finally gives beta carotene. So this is very important because after this story, we will also I will also explain you our own work with the tomato, where we improved lycopene content using genome editing. Okay, so next slide. This is it worth golden rice? Just look at this slide and let me know whether it is worth to produce golden rice. Oh, is it visible to you, or I need to read it? Okay, this is sweet potato that gives 173 microgram per gram provitamin A, and this is carrot which gives 125 microgram per gram, and this is golden rice which gives 3.5 microgram per gram. So now question arises, if we have such excellent sources already available, that is sweet potato or carrot, why we stuck with this golden rice? Mm-hmm, mm-hmm, mm-hmm. Yeah, if people eat daily, right? The uh, the target population that eat rice daily. And second important thing, if we have golden rice, that is one time, right? Once we created golden rice, we can harvest several generations. The seed will be there. Right? It is sustainable solution. And this is not something like a uh, replacement. This is complementary. If you are eating golden rice, you can also eat sweet potato, or you can also eat carrot. It is not something, not like a replacement. It is a complementary. That's why it is very important to have a golden rice. Okay. So we have another example, that is golden banana. Right. This technology was developed in Australia, and now in India also we have several labs working on golden banana. So Rajiv Gandhi Center for Biotechnology in Kerala, they are prominently working to produce a golden banana. So now the same question, is it worth to have a golden banana? Is it worth to have golden banana? Because rice is a staple food we consume daily. So what about golden banana? Yeah, so equally wherever banana is available, carrot is also available, sweet potato available, papaya available, mango available. Okay. The list which I have shown in an earlier slide, banana is a costlier than sweet potato, right? Banana is a costlier than the carrot. Banana is a costlier than the papaya. So whether it is worth or not. So I need a valid argument to support the golden banana. If we talk about nutrition, we cannot say that the banana has a more nutrition compared to sweet potato or papaya, right? Because when you compare the provitamin A content, here golden banana has a 5 microgram per gram, or sweet potato has a 173, which is almost like 50 times more. Right now, here we cannot give that argument that is like a rice, it is not a staple food. Second, if we are targeting school children in a target population, the transport of rice is very easy, storage of rice is very easy to use in a midday meal program. But when we talk about banana, if we say banana is like more stable, it's not quickly perishable, but equally the carrot and sweet potato is also right. So sweet potato and carrot is equally convenient to transport. Okay. Banana we don't need to cook, right? Banana we don't need to cook. We can just distribute in a school and they can eat, right? But it is not a valid argument because carrot also we don't need to cook, right? Okay, but most of the population like they don't cook the carrot, right? Or for banana also we can make a sabji of banana. That is the options are there as well. So okay, most of the people prefer banana over the carrot or over the sweet potato. But it is always a question whether they will prefer the golden banana or not. That is strategic one, right? So you can think there is no definite question, certain question answer for this question. So next thing, this is also important. Here, banana is a white banana or red banana is there already, and we are trying to make a transgenic to make it a golden. And there golden tomatoes are there. We are trying to make it like a red, right? So where we already have nutrient, we are trying to make it something else. Like we wanted rose to be green and then the parrot to be red. So it is something like that. Okay, next. So now we will ex- uh, explore the area of transgenic crops, like how these transgenic crops are very important for the biofortification and what are the pros and cons with this. So there are several different transgenic crops are being grown worldwide, and major are the canola, soybean, maize, rice. Those are the major crops which are being grown worldwide as a transgenic mix. Okay, and these are the countries. So US is the largest one which grow transgenic crops. Then the Brazil, Argentina, Canada, and then finally India. In US, it is very easy, or their regulations are not very strict to adopt any transgenic crop. So most of the transgenic, whichever are being grown worldwide, those are grown in US also. That's why US has highest acreage of the transgenic crops. In India, India is also having very high acreage, but that is only with the one crop, that is Bt cotton. In India, we have only one transgenic. Okay. So next slide. So these are the crops like in USA that has a 75 million hectare. They grow maize, soybean, cotton, canola, sugar beet, alfalfa, papaya, squash, potatoes, and apples. All these are the transgenic. And for the transgenic also they have several different traits for transgenic like herbicide tolerance. They have insecticides like Bt in different crops. They have also transgenic for the improved oil content and they have transgenics for the improved shelf lives. So these are the major traits. Okay. Then we already have discussed that, right? We have only one transgenic because if there is a concern with the regulatory issues, there is a concern with the environment, right? We cannot easily release any transgenics because we cannot take it back easily. And that's why it is very important to have an alternative approach that is genome editing in plants, which will produce a desirable trait but without having any transgene. And these are the transgenic genomic uh, plants already released, like in the maize, ALS2 gene is edited that gives herbicide tolerance. This is PSY gene that improve the phytoin content. So there are several different crops are already released commercially or in the step of releasing. Like in, recently last year, GABA rich tomato was released in Japan. So many of these crops are at the stage of releasing for the commercial cultivation, or some are already released. So next one. So we already have seen the golden rice with the transgenic approach, where genes from maize or daffodil was used, or Erwinia bacteria was used to create a golden rice. But without transgenic also we can make a golden rice. And for that we can use genome editing, which can change this OSOR gene, that is O is for Oryza sativa, and OR is for orange. This is the same gene which is used to create a orange colored cauliflower. This orange gene was targeted to create orange colored cauliflower. And in rice, that gene is present. And if we modify that gene, if we make any deletion in that gene, the rice grain become orange in color. And that will have a more provitamin content. That is beta carotene, right? And that we can do with the genome editing. So this is the gene, and these are the target site with the for the genome editing. With CRISPR-Cas technology is one of the method which we use for genome editing, and that can target to this position in the gene. This is the gene. These boxes are exon. This is intron. So first exon, second exon, third exon. If we make deletion in this gene, that create a orange color callus. This callus is orange color. That means that has a beta carotene, right? And that's how we can use this genome editing approach without having any transgene and get the similar desired improvement in the traits. Okay, next slide. So, tick time tick chal raha hai. Okay. So, some popular examples are there. So next slide. So, this is maize, and which has a high amylopectin, and this was created with the genome editing. And this is important to provide a quick energy. If we have high amylopectin, that will be providing quick energy source, like for the soups, maize corn soup. That is important. Even to make a high muscle mass, this high amylopectin is important. I think you know better than me because most of you are MSc in nutrition. Okay, so next one. This is camelina, which was developed with the genome editing. Here, three different genes were targeted to improve the oil content of the camelina. Next one. This is soybean, where this DRB2A and DRB2B, these genes were targeted to make it drought tolerant. So this soybean required less water compared to the wild type. So this become a more drought tolerant. Nowadays, it is very important because of the unpredictable weather, because of the climate change, the frequency of getting drought or flooding spells are more frequent, and for that, such crops are very important. Next one, this is a mushroom, because in mushroom, browning is a major issue. Whenever we harvest mushroom, it gets quickly browning. So this PPO gene was targeted, polyphenol oxidase, so that is enzymatic browning. PPO is a polyphenol oxidase that make this apple or potato or this mushroom brown. So if we remove that gene, that will stay more for more period. That can improve the shelf life of this mushroom. Next one. Okay, so this is the there are several different methods of genome editing, and this cluster regularly palindromic reason, that is CRISPR. And CRISPR mediated genome editing is the very convenient. So nowadays that get a boom. Like most of the uh, institute, they are working with genome editing, and they are exploring this research advancement in a CRISPR based genome editing. So next slide. And this is very common picture. You might have seen in most of the biotechnology department, or maybe in another department, life science department also. This is a picture of Emelina Chape and Jennifer Doudna. They got Nobel Prize in 2020 for the development of this CRISPR-based gene editing technology, and this was the quickest Nobel Prize. And in every presentation, I ask the same question: what is unusual in this one? Yes, that is on the lighter note. The two females are working together to develop such fantastic techniques. So it is very uncommon, but this is in a lighter note. There are female scientists, they collaborate to each other. This is not that much that unusual to highlight. Okay, next one. So this genome editing story started long back. Although this Nobel Prize were the quickest one, the Jennifer Doudna and Emmanuel Shafe, they got the quickest Nobel Prize. Usually people get Nobel Prize when they discover something after 1 or 2 decades, then they get the Nobel Prize. The same thing happened for this one also. All the three Nobel laureates in physiology and medicine, they are above 60 years, right? So this was the very quickest, because this technology got a boom and people have trying to unlock the potential of genome editing. There are several ways, several methods are developed based on the CRISPR-Cas. But the story started long back in 1987, and then in 2000, this Emiliana Shape and Jennifer Doudna, they pick up this area, and then they succeeded to develop the genome editing. So next slide. Okay, there are several different methods of genome editing, but we will, for today's presentation, we will focus on the CRISPR-Cas based method. Next one. Okay, next. Okay, these are the four major techniques which we can use for the genome editing. First one is the meganuclease. Here we can, first we need to design a meganuclease enzyme which can have a cut at a specific location in the genome. Second technique is zinc finger nuclease, then the TALEN, and the latest one, the CRISPR. So we will go through one by one. Just a quick next slide. So this is zinc finger. So zinc finger is a transcription factor which can bind to specific sequence in a DNA. So we can design such a zinc finger which can target to any specific gene. So we just need a different zinc fingers, combine those together, and then we can target to any specific gene. Suppose I wanted to target to lycopene beta cyclase gene in a tomato. So this lycopene beta cyclase gene in tomato is very important. That gives a red color to the tomato. Right? The red color in tomato is because of the lycopene. That's why we call it as a Solanum lycopersicum, right? So second one, this is the TALEN. TALEN is also the same, where we design a TALEN which can target bind to a specific sequence. And with TALEN or gene finger, we also attach one endonuclease, that is fork one, so that we can make a double stranded break in the DNA at any specific location wherever we want in the genome. We can design that, and then this fork can make a double stranded break. Okay, next one. Next. Next. Okay, now this is CRISPR. So what is CRISPR? CRISPR is a clustered regularly interspersed small palindromic repeat. So here you can see in the bacterial genomes, these repeats are present. Like this is palindromic. You can see CAATAA from this side also, CAATAA, CTT, this side. The sequence is same here also, and in reverse complementary direction it is same. That's why it is a palindromic, right? And this is very small sequence. And why clustered regularly? Because those are present at a regular interval. That's why we call that as a clustered regularly small palindromic repeats, because they are completely repeated one after another one, right? So next slide. So the question was that these repeats are present in a bacterial genome, sometime in a thousands in number. So scientific community, specifically working with the microbes, they were focusing, trying to answer the question why these repeats are present in a bacteria. And because the prokaryotic genome is compared to more compact compared to eukaryotic genome, and having such a repeat was surprising, right? If such repeats are present in almost more than 1% of the total genome, that was surprising. And then Francis Mujica, he started working on these repeats in 1980s, so early. He did his PhD on this CRISPR repeats. Then his labs worked almost 2-3 decades on the CRISPR repeats. Next slide. And he first discovered this CRISPR repeats in a bacteria. Then he first showed that there is a PAM domain, protopiscer adjacent motif, which is required for the functionality of this CRISPR system in a bacteria. So next one, I will not go deep, but this is important to understand the CRISPR mechanism. So this is in bacteria. So in bacteria, the bacteriophage, whenever infect the virus, which can infect the bacterial cell, whenever that infect the bacteria, that release the genome into bacterial cell. And this genome get chopped into small pieces, and some of the pieces will get incorporated in between these repeats. These blacks are the CRISPR repeats. And in between two repeat, the unique sequence that is from the different virus. So this blue one from this virus, this one greenish one is from the another virus, this sky blue is from the another virus. So basically, these repeats are present in bacterial genome, and in between this repeat, there is a unique sequence that comes from the different viruses, right? And once this fragment is inserted into this repeat, that get transcribed in a long RNA that we call as a polycystic RNA, right? In prokaryotic system, the different genes can get transcribed together as a polycystonic unit. In eukaryotic system, most of the gene are like single promoter, single gene, single RNA, right? Only tRNAs, those get transcribed as a polycystonic unit. So once that get polycystonic long RNA, every single unit gets chopped separately. Single unit means this fragment from this virus and one repeat. This is one unit. Second fragment from second virus and this repeat is second unit, third unit. Likewise, thousands of units. They get chopped into single piece. After chopping into single piece, there is one another gene present in bacteria that is known as a Cas, right? CRISPR associated protein. This Cas9, there are several Cas, so this one is the Cas9. Then this Cas9 make a complex with the single unit. Single unit means this unique sequence of virus and repeat. That they make a complex. And then wherever this small RNA1 unit has a complementary sequence, that will go and bind there. For example, the same virus infected the bacterial cell again, this sequence has a similarity. So this unit will go and bind to that virus genome because of the complementary sequence. And then this Cas9 enzyme will make a double stranded break. Since this Cas9 is making double stranded break to the virus genome will not replicate, and that will provide immunity to the bacteria. That means if any virus piece is get incorporated between two CRISPR repeats, next time the same virus cannot infect to the same strain of bacteria. That is exactly like how immune system works in our body. Whenever we get a COVID vaccine, then the COVID virus will not infect us, right? In the same way, in a bacteria also, this CRISPR system work as an immune mechanism. And once this mechanism get understood by Emilina Chape, Jennifer Doudna, then they use that mechanism to create a tool. This is exactly the same how we use EcoR1 enzyme as a molecular scissor, right? Most of you already know EcoR1, right? That is from the E. coli R1 enzyme, which is a endonuclease, right? And that can make a double stranded break at GAATTC. That is the site, recognition site for EcoR1. But in case of CRISPR-Cas, we cannot change recognition site of EcoR1. That will always get at GAATTC. But in case of CRISPR system, if we change the sequence of this single unit RNA, this Cas9 and this complex will bind at that same sequence. That means in a genome, if we take any sequence from any gene, we can target our Cas9 at that location. Suppose I am working with lycopene beta cyclase, I will take a 20 nucleotide sequence from lycopene beta cyclase gene, and then I will use that as a guide RNA for this Cas9. And then this Cas9 will make a double stranded break in a lycopene beta cyclase gene. So this double stranded break we can use in multiple ways. First, we can just make a double stranded break, and inbuilt mechanism in every eukaryote or prokaryote that will rejoin those ends. That is DNA repair mechanism. But whenever this cut end joined back again, that is always faulty. Sometimes there is one nucleotide less, sometimes there is five, sometimes hundreds of nucleotide less. Even if there is a one nucleotide less, that will create a frame shift. If in any gene, if we remove only one single nucleotide, that will change entire protein. That will affect the functionality of the protein. The function will be lost because of the even one nucleotide. If we remove, the codon frame will change, right? And the protein will completely destroy. So making any protein non-functional, it is very easy. That is the first way. Second way, we can insert any gene, any nucleotide sequences between these two double stranded break. If we provide any donor sequence that we can incorporate. So now this genome editing technology advanced in such a way we can make any changes in a sequence. We can insert sequence, we can delete sequence, whatever changes we wanted to do that we can do with the CRISPR-Cas. Okay, next slide. Okay, we can see this again with step by step. First step, when bacteriophage infect to the bacteria, the genome get chopped into small pieces. Some of these pieces will get incorporated in between this CRISPR repeat. Second click. Then this, whenever there is second time infection, this will get transcribed into a long RNA and chopped into single unit. So this is one unit. Again click, then this will bind to the complementary site, and then again click, this will make a double stranded break so that the virus will not replicate. Next slide. Whenever it create a double stranded break like this, the Cas9 will create double stranded break at the target site. Next slide. And then this will get a repeat, like this is double stranded break, click, and that will get repaired with the DNA repair mechanism present in any living cells. Then we can use this very simply, like we can clone this Cas9 and this guide RNA for whatever gene we wanted to modify their complementary sequence in a plasmid vector, and then transform this vector to the plant and then create a transgenic plant. And in transgenic plant, we can sequence it to see whether there is any modification, desired modification happened or not. Next. Next. So back. There are several different methods with the CRISPR-based genome editing in plant where we can target multiple genes also, right? We can, with genome editing, we can change any single gene. We can target one gene as well as we can target multiple genes. So when we wanted to target multiple gene, that is relatively more complicated compared to single gene. And for that, several methods available, like individual promoter, CS54. These are the methods. I will just skip those, and then we will say more focus on the tomato story. Next, 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 next. Okay. So why multi-target genome editing is important? Because some in specifically in crop, some crops are having polyploidy genome, like wheat is a hexaploidy, right? And several brassica species they are having different ploidy levels. So there, if the ploidy level is more, that means every gene has a multiple copies. And if we selectively wanted to edit any specific copy, we need to target a more number of genes. Then the polygenic traits. Not all the traits are monogenic. Some trait, if we wanted to improve some trait, we need more number, like more than one gene. Like as we have already seen in golden rice, we need the three genes, two from the daffodil, one from the Irvine, right? So most of the time we need to modify multiple genes, and for that this multi-target genomic important. Next one. Okay. So one another very interesting application of this multi-target genome editing, we can use multi-target genome editing to have a de novo domestication. Okay. What is de novo domestication? Usually, how domestication happens? Like for rice, whenever first time someone found out that in a wild forest or wild habitat, one rice has a bigger size of grain, and then someone collected it and started growing it. That's how crop get domesticated slowly, right? But if we wanted to domesticate a new crop, we can use this multi-target. For example, here, this is Solanum lycopersicum. And for the conventional breeding, if we wanted to bring salt tolerance, cold tolerance, drought tolerance, we need to cross this Solanum lycopersicum with the wild tomatoes. And then we can have this all tolerance in a high-yielding variety. So with the every crossing, there is a unwanted genetic linkage drag, right? When we cross the high yielding variety to wild variety to bring in salt tolerance, that will also hamper the quality taste of the high yielding variety. That is the drawback with the conventional approach. But if we converted directly this wild type to the cultivated type, this is Solanum galgapinium. So if we target these 6 different genes, that will be exactly like our domesticated tomato. So these are genes like a self pruning that change the plant habit. Then this CFW 2.2, this is proved weight. Then this is local number that will increase the local number. So see, if we target 6 different genes, that will convert this wild type to the domesticated type. And then we can easily use that for the breeding purpose. Okay, maybe little complicated for you. Just if you understood it, it's good, but if not, it's okay. So next one. Okay, so our lab, means my lab as well as Dr. Humira's lab, we work together with the genome editing, and we work with the 4 different crops. We work with tomato, mustard, rice, and soybean. These 4 crops we are trying to improve with this genome editing technology. And after this class, we can visit to our growth chambers to see how exactly we do this genome editing. Okay, next one. So this is, we have already seen this one, right? Very simple. We just clone the guide RNA and Cas9 in the vector, and then we transform this vector with the agrobiotic bacterium mediated transformation, and then we grow the transgenic plant and then sequence that gene to see if there is any desirable changes or not. नेक्स्ट वन, कितने लोग जिंदा है अभी? चलो ठीक है, काफी नंबर है, ठीक है, ठीक चल रहा है. Okay, next one. Okay, this is the one method where we use CSY4 and like we use one promoter, first guide RNA for one gene, second guide RNA second, third guide RNA for third, fourth for the fourth one. The four guide RNA for four different gene, and every guide RNA is separated with this CSY4 site. This is the same. This is the gene from the bacteria. This is the same from the same system from CRISPR-Cas. In bacteria also, I have shown you that the long polycystonic RNA get transcribed first, and then it gets chopped into single unit. And that chopping mechanism is because of this CSY4. And we use the same CSY4 here in the system to separate four guide RNA. That will chop every single unit separately. So this is first unit for first gene. This is for second gene, third gene, fourth gene. So we can target up to 6 different genes with this multi-target system. Okay, next one. So we started working with tomato because everyone knows that tomato is very important worldwide. Any country that grows tomato, whether it is in Africa, whether it is in Europe, whether it is in Canada, so tropical, temperate, everyone they grow tomato either in open field or maybe in greenhouse, right? But every country they grows tomato. It's very important crop as a food as well as an industrial crop also. We consume tomato raw, we consume tomato as cooked, we consume tomato as a processed food. That's why tomato is very important, and that has a major issue, that is the shelf life. Shelf life is very short. That's why sometimes it is in India like a 10 rupees kg, sometimes it is like a 200 rupees kg. And that's happened because we cannot keep tomato for a long time. That's why we targeted to improve the tomato shelf life as well as other traits which are important to improve the nutrition. Okay, next slide. Okay, so shelf life is a problem. Next one. Already several gene people tried, but we selected peptide liase. Next one. Next one. Okay. So in this project was in a two step. In first stage, we targeted three genes. The cell pruning 5, that is peptide liase and lycopene beta cyclase. This cell pruning, this gene help to change the plant architecture. So this is indeterminate plant type. If we mute this gene, that will create more compact plant, that is determinate, or we can make a double determinate also. Okay. Second is pectin liase, that improve the pectin content in tomato, so that fruit cracking will reduce, and we can keep tomato 2-3 weeks more than the normal tomato. The edited line, the tomato with the edited line, we can keep for 2-3 weeks more. Then this is lycopene beta cyclase. This is beta carotene pathway, and if we knock out this lycopene beta cyclase, that will improve the lycopene content. I have already shown this pathway to you in a golden rice, right? We will see it again in concern with the tomato. Next slide. Okay, so this is carotenoid biosynthesis pathway, and here actually in golden rice also, and in case of tomato also, we are targeting here. The next slide. So this is the lycopene converting to beta carotene. And if we knock out this gene, lycopene beta cyclase, that will stop the conversion of lycopene to beta carotene. Right? We use this lycopene beta cyclase in case of golden rice, where we produce this beta carotene. In golden rice, we wanted to improve the beta carotene because it is a provitamin E. But here, we stop the conversion of lycopene to beta carotene. So, is it worth doing this or not? क्वेश्चन क्लियर है? गोल्डन राइस में हम लोग ट्राई किया कि बीटा कैरोटीन कैसे बढ़ेगा. We use the transgenic approach to improve the beta carotene, and we targeted the same pathway. There, naturally, beta carotene production is not there. We use gene from the daffodil or irvine to create a beta carotene. But here, naturally, beta carotene produces, but we are stopping conversion of lycopene to beta carotene. So my question is that, is it worth or not? एग्जॉस्ट हो गए या अभी जिंदा है सब लोग? जिंदा है, चलो ठीक, बताओ.
सपोज सर आपने टारगेट लाइकोपिन को किया है तो फिर बीटा कैरोटीन को इनहिबिट कर दिया सर आई थिंक इट्स ए गुड आइडिया
Any other opinion? For time, save time for what? Mm-hmm. Okay. Okay. Actually, beta carotene is very important because this is provitamin A, but lycopene is also very important. That is also having a very great nutraceutical value. For beta carotene, as I have shown you, sweet potato has 173 microgram per gram, and then the carrot has almost 100 microgram, right? So there are several sources, but for lycopene there are very limited sources. So till if we are sacrificing with the beta carotene contained in tomato, it is good idea to stop this conversion because of the two factors. One is lycopene is also nutraceutically very important. We have very limited sources for the lycopene. Second, if we improve the lycopene content, the tomato will get a better price because in a market people prefer more red tomato over golden or yellow tomato, right? That's why it is good idea to improve the lycopene content. Next one. Sorry. Yeah, that's why that has a better nutraceutical values. So we use the same simple technique, just clone the target site of lycopene beta cyclase, pectate lyase, cell pruning 5, make a construct, make a transgenic, and then evaluate it with the sequencing whether we got a desirable changes in our lycopene beta cyclase gene or not. Next slide. Next one. Okay. So these are the more the practical thing. First, we need to select appropriate target. So the tomato genome sequence is publicly available. We use the tools to identify the gene, and then we prepare to target. If we wanted to knock down the function of the gene, we prepare to target the first exon. If even if one nucleotide change is there, even if one nucleotide deletion or insertion there, that will destroy the function of the gene, right? So we use different tools like CRISPR-P, and then there are more thousands of tools are available to identify the target. And once we decided to target, we clone that sequence to our vector for transformation. Next one. Okay, so this is case of like in beta cyclone. Okay, whenever genome sequence is available in a public domain, that is for a specific variety. Not all the variety have a publicly available genome sequence. So in India, most of the lab, they use Pusa ruby variety of tomato for transformation, right? So earlier it was thought that the Pusa ruby is easier for transformation, but the genome sequence available publicly, that is not from the Pusa ruby. And if you select a target based on the available genome sequence, that may differ from the variety which we are using for transformation. And for that, we need to sequence gene in our variety, and then the confirm the target sequence is the same, right? Second one. And then we prepare construct like use one promoter for Cas9 gene, and then we have second promoters, or sometimes with the same promoter with the cleavage site, we use a guide RNA and selection markers. Okay. We will just skip the more biotechnological practical details. Okay, next one. So this is the multi-target construct which has a different guide RNA for 3 different genes. We clone in a one single plasmid vector. Next one. So for transformation, we use 4 different tomato variety. Pusa ruby, as I mentioned, that in most of the Indian lab, they use a Pusa ruby for transformation. And then we use Artha Vikas, this is high yielding variety. Pusa early dwarf, this is very short variety. And in our target gene, we selected 3 target genes. Cell pruning 5 that changed the tall tomato varieties to short one. That is actually changing architecture based on the determinant indeterminate, like that convert indeterminate plant type to determinate. Indeterminate means the plant continuously grows. Even if there is flowering, that will again grow. There will be fruits also at ripening stage. There will be flowers also at the same time. So that will continue. Double determinant or determinant means that will get flower once and that will get fruit once, and then the life cycle is over. So for that we selected this Pusa lead to see whether the same gene, what effect we can get with the same gene. And then this is Solanum pervinum. because here we are targeting 3 different genes, and I have already shown you how we can use this multi-target genome editing for the de novo domestication. So here we also wanted to see whether we can domesticate this Solanum perennium, and for all this purpose, the most important thing is the protocol for transform all different varieties. As I mentioned you, in India, most of the lab they use Pusa Ruby because it is easier for transformation. So we develop first protocol which can transform all different varieties, including the wild type also. So now we have protocol which we can use for any tomato variety, and that gives us flexibility because we can now use that protocol directly for the high yielding variety. Otherwise, if we don't have such protocol, then we need to transform first one variety and then we can again take it to the high yielding variety by conventional breeding method. So that is more time taking. So next slide. Okay, next slide. Oh, okay. So after transformation, when we sequence the transgenic plant, which express this Cas9 and guide RNA, then we sequence our target gene, and then we found that the desirable mutation is there. Next one. Okay. So here some of the detail of some of the lines for 3 different genes. So this is first gene, lycopene beta cyclase. This is control sequence. Then this is AV means Arcavica. As I mentioned that we targeted 3 different genes in 4 different varieties, right? So first one was Arcavicas, second was Pusa ruby, third was Pusa early dop, and fourth one was the wild type tomato. So the numbering is like AB means Arcavicas, PR means Pusa ruby, PED Pusa early dop, S means Solanum pyrenum. And this 64 means 64 number of plant which we grown with the right. So they got a different deletion, 11 base pair deletion, 5 base pair, 17, 10 base pair deletion. So with all these deletion, the function of this gene will destroy, right? So likewise, we got a deleted deletions in all for all 3 different genes with all 4 different varieties. Next one. Oh, so there are steps like first, whenever we grow plant with the transformation, we call that as a T0 stage. And when we get a fruit to the T0 plant, when we harvest it and then plant it again for the next generation, that is T1, right? So in T1, we again confirmed it with all different lines. So at present, we have about 200 genome edited lines in 4 different backgrounds and with 3 different genes. So there are lines with all combinations like some line, like for example, in Pusa, Arcavicas, that is variety, that has some line with only one gene mutated, that is lycopene beta cyclase. Some line has a lycopene beta cyclase as well as SP5G. Some line have all 3 gene mutated. So when we use this multi-target genome editing, that help us to create a catalog of lines. Catalog of lines means that will have all combination of mutations. Some line will have mutation in only one gene, some will have 2, some will have all 3. So all possible combination. That help us to select more desirable line in terms of production, in terms of nutraceutical value. Okay, so next one, just we will skip this. Next. Okay, so this is how what phenotype we achieved with. This is example for only one gene, that is SP5 gene. So here this is Pusarli dot SP5G. That means this is Pusarli dot. When we mutated this SP5G, that become this one. This is Pusa ruby. Naturally, the wild type Pusa ruby is an indeterminate plant, so that grows very tall and that grows continuously. When we mutated this SP5G, that become very short and determinate, right? So that provide advantage when we grow tomato in a field. Like if we are growing determinate variety, that will get flowering at one time and we can harvest once and the field will be vacant for the next crop. But in case of indeterminate, we need to continuously pick the tomato fruits every week. So that is more labor intensive, as well as we need to take those tomatoes to market every week. So that is not advisable to use indeterminate variety in a field. But if you are growing those tomato varieties in a greenhouse, then usually people prefer to have a variety which gives continuously fruit, like indeterminate, right? So here, this figure also make it clear why we are targeting the same gene in 4 different genetic background, right? Suppose in case of Pusa ruby in wild type, normal Pusa ruby variety, there is typical pattern. We get a flower first, then first internode leaf, second internode leaf, third internode leaf, and then flower. So flowering will be always like first flower, one leaf, two leaf, three leaf, and then flower. So three in between internode. When we mute this gene, that become flowering become like a first flower, one internode, second internode, and flower. One leaf, like there is a more frequent flowering. Like instead of three leaf, there are only two leaf. That case is in a Pusa ruby. But when we mutated same gene in Pusa early dot, this one, Pusa early dot, there is like a leaf, flower, leaf, alternate, right? Because Pusa early dot, it is already dot variety. That is determined, already determined. When we knock out the same gene in that variety, the flowering become like with every internode. Like if there is a in an internode, if there is a leaf, there is a flowering also. So every internode got a flower. Same gene in two different genetic background gives a different result. So all different four different varieties, we got a different phenotypic expression of the same gene. That's why it is very important to also consider the genetic background. We cannot expect the same result with the every genetic background. Okay, next one. Now we can speed up. So this is the result with the pectide lies. The bottom line is edited, the top line is for the wild type. And that improved the shelf life by 2 weeks. Next one. So this is the fruit cracking. If pectide lies, we need more pectide lies, that will improve pectin. That will also reduce the fruit cracking, because whenever we transport tomato from field to market, we need to go with the truck load, and that damages many fruits. So that also improve the fruit firmness. Okay, next one. So this is lycopene beta cyclase. When we knock out this lycopene beta cyclase, lycopene content improve. And to like we can directly, it is visible. You can directly see through the edited tomatoes, those are more red. But to have a quantifiable data, we use HPLC. Here you can see this is the edited one that has almost twice lycopene content compared to the white. Okay, next one. Apart from them, when we edited SP5 gene, the gene which make it determinate, that also improved the root architecture. This is wild type tomato. You can see the shoot size in all these conditions are almost same, but the root is almost 3 times longer in case of edited lines, right? So when we modify the plant architecture, shoot, whenever we modify shoot, root will automatically get modified. Okay, next one. Now we are at the second stage. As I mentioned that in tomato, we are working with the two-stage project. First stage, we selected 3 different genes and we developed 200 genome-edited tomato lines for 3 different genes combination and 4 different genetic combinations, genetic backbones like Pusa, Ruby variety, Arlivica, Arcavicas, Pusa Arledorf, and then Solanum perulum. Okay. Then the second stage started, like my present PhD student Badal Mahakalkar, he is working on this project. So here we are trying to incorporate GABA and provitamin D. So we wanted to improve GABA content in tomato and provitamin D also, because GABA is important to regulate the hypertension, and in India it is very frequent problem, right? Almost everyone above 45 age, half of the population is taking medicine to control the hypertension, the blood pressure, right? And for that, this is GABA rich food is very important, and there are very few food crops are available which provide us GABA. And among that, tomato is there. But with tomato, green tomato has a GABA content, but when it get ripened, the GABA get degraded. The ripened tomato, they don't have a GABA. Green tomato, they have a GABA content. But we use ripened tomato instead of the green tomato. So we decided to use this approach just to stop the degradation of the GABA content. And that is because of one gene, that is GAD3, that has the autoinhibitory domain. Right? If we remove that domain, that will continuously synthesize the GABA. And for that, genome editing is very easy approach. Instead of selecting first exon, here we selected last exon just before the autoinhibitory domain, just to remove the autoinhibitory domain so that the GABA production will be continued in a ripening tomato. Next one. So this is the GABA shunt pathway, and here you can see this is the GAD3 gene, and this is autoinhibitory domain. So we targeted here just to remove this domain so that the GABA will be continuously produced. Next one. Is it okay or need to speed up? भूख लग गई? 1 बज गए? Okay, chalo, speed up kar and then we targeted this 70 hydrochlorostrol just to improve the vitamin D. Next one, next. Okay, and then we have some other projects coming up. One is this earlier the DBT sanctioned this project in 2025 year back, but they never released the funding for this project. In this project, we wanted to improve the bran oil content in rice, which is one of the largest underutilized commodity. We produce a lot of rice bran, but we use very like less than 10% of the bran for oil production. And you might have already seen the rice bran oil in the market. Right? Most of the shops they have this rice bran oil. Next one. Next one. Okay, this is the pathway where we are targeting to improve the rice bran oil. Here, fat to gene which convert oleic acid to linoleic. If we mute this, oleic will be improved, and this is triglycerol. So, if we targeted this triglycerol lipases, that will stop the hydrolysis of triglycerols, so that the bran oil quality and quantity will be improved. Next one. So, we have this project with the Pune University. Next one. Okay. Another project we have to improve the rice breeding because rice is a self-pollinated crop. And if we wanted to grow hybrid rice, like in China, they are about like 50% of rice area is with the rice hybrid. In India, it is less than 10%. In India, we don't have hybrid varieties of rice. And to improve that, we have this project. Next one. Next one. Okay. In this project, we are trying to make a rice line which has a two male sterility system. For male sterility system, there are two options: photoperiod sensitive male sterility and thermosensitive male sterility. These are two options in rice. So we are trying to combine both options with the genome editing, so that will be more uh, sure, more I think stable male sterile lines. Next one. Next, okay, next one. Okay, this is also a new trait. In rice, usually we have one fertile spikelet and two sterile spikelet. In rice, basically, in rice, there is one fertile spikelet and two sterile spikelet. Naturally, this is the condition. But if we target this combination of genes, that will create three fertile spikelet. These two sterile spikelet will convert into fertile one, so that for every spikelet there will be three grains instead of one. Okay. Next one. So multi-target generating soybean. Next. So as I mentioned earlier, my lab worked with the silicon, right? And we wanted to improve the silicon content in plant to make a plant resistance against biotic stress, as well as silicon is beneficial for the human bones, right? And for that, we already identified gene with the different techniques like QTL mapping, GWAS, then genome sequencing, then association mapping, all those things. And we reached at the stage where we can use this genome editing. So in this, here we targeted 6 different genes, every gene with the 2 targets, and then we achieved the desirable mutation in soybean. So next one. This is the deletions which we obtained in soybean. Next. Then the Dr. Humera's group is working with the food grade soybean because in India, we are the largest vegetarian population. Many of us take non-veg also, but it is not frequent, once a while. But most of the population is dependent on vegetarian source for the protein. And for that, it is very important for us to have a food with the protein rich. And soybean is one of such of source which we can explore. But we don't like the taste of the soybean or the flavor of the soybean that has a beanie flavor. And to remove that beanie flavor, Rumira is working with that. They are targeting several different genes in which LOX gene is there. If we target that LOX, that will remove the beanie flavor. Another, they are targeting with the fatty acid biosynthesis pathway, FAT2 gene. If we target that gene, that will improve the oleic content, right? Next one. Next. Okay. So to identify that gene, Dr. Mira has a very large soybean population, diverse lines, and that got evaluated at different locations. Like here you can see the field trials in Srinagar, then YS Parmar University in Himachal, PDKV University in Akola, Maharashtra, Indian Institute of Soybean Research at Indore. So several different locations, those lines grown and then identify the genes which we can target with the genome editing. Okay, next one. So actually, Dr. Humira's group and Dr. Humira has expertise with all these techniques where we can use like whole genome sequencing or genotyping through genome-wide association study to identify genes. And once we identify gene, then only we can use that information with the genome editing. If we don't know genes, we cannot use genome editing to target anything, right? Next one. Okay, next. Okay. And I must thank for all this work to different funding agencies and my student, because this is all the work is with the students. They did their PhD. They work day and night in the labs uh, to get this result. So Sanskriti was my first student. She worked with the genome editing in tomato, and she is presently at the University of Camping in Denmark. And no, she moved again because she got all the fellowships. She got Fulbright, uh, Curie, she got Gated. So all fellowships she has. So she has very freedom to move to different labs. And the second student, Yogesh, he worked with the molecular dynamics in aquaporin rice, and we are targeting that aquaporin to reduce arsenic content and improve the silicon content. Next one, then Ruchi Bansal, she worked with the rice haplotyping, and she is a data scientist at Amazon. Then Surbhi Kumawat, she worked with the aquaporin improved development in bottle guard. She is postdoc at Kansas State University. Vandana worked with Mungbin aquaporin. She was here also for short period as a NPDF, and then she moved to the Western Virginia State University. And then Gaurav Raturi, he worked with the silicon solubilizing bacteria. As I mentioned you, one of the approach which we can use to improve the biofortification, that is the microbial applications. So he worked with the silicon solubilizing applications. And then he's doing postdoc at Texas Tech University, USA. Next one. Now at present, Mukesh is here. He is working with aquaporins in onion. Badal is working with multiplex genome editing in tomato. He is trying to improve the GABA content as well as vitamin D content in a tomato, which already has a high lycopene content, small plant type, and a longer shelf life. We already edited 3 genes. Now Badal is adding 2 more genes. That is called as a gene stacking. We are stacking desirable genes in a same genetic background. Then Pragati is here, right? Oh, Pragati changing the slides. Pragati is working with one of the subfamily of aquaporin, that is G, uncharacterized interstick proteins. Then Pawan is working with Pawan is actually working with so many different projects. He is working with the genome sequencing of insect. He is working with aquaporin. He also working with developing a microbial consortium. So he isolated more than 200-300 rhizobium, and he is looking into rhizobium having a different characteristics. Some rhizobium they fix nitrogen as well as they solubilize phosphate. Okay. So Gitanjali is here. Gitanjali is working with the metalloid transporter selenium basically, and Akash is also here. He is working with the metalloid transporter aquaporins. Okay, thank you. Any question?
Good afternoon, sir.
Good afternoon.
Thank you for the very informative session. And my question is, when pearl millet is fortified with iron, so does it cause any change in protein content?
Oh, I don't know exact answer, but I guess there will be very less or almost no change for protein. I'm not sure about this.
Okay. Thank you, sir.
But for rice, zinc rice, when DRR released, I think 4 different varieties of rice. With the zinc rice, there is no significant change with the yield as well as the protein content, because rice has almost 5 to 8% of the protein, and the protein content fluctuate more based on the fertilizers, like if we use urea or nitrogen fertilizer more in rice, the protein content also fluctuate. But with the zinc fortified, there is no not significant difference.
Okay, thank you, sir.
Yeah, actually, my work is not completely focused with the biofortification in cereals or maybe in millets, but rather I am using this genome editing technology to translate the available knowledge into developing a product. Like in case of tomato, we are targeting the previously characterized gene like lycopene beta cyclase to improve the lycopene content. Or we are for GABA also we are targeting like 7-DHC gene or GAD3 gene for the provitamin D and GABA containing in the tomato.
Thank you, sir.
बस इतना ही क्वेश्चन नहीं है कुछ थैंक यू
Hello, sir. Sir, I want to know that how genetic engineering is differ from conventional breeding to improve nutritional content in crops.
Okay. Actually, I was also expecting the question like when I have shown you that we first make a plasmid vector and then we transform to the plant to have for the genome editing. In plasmid vector, we have guide RNA as well as Cas9 gene, and then we make a transgenic and then we sequence the target gene to see if there is any modification. So whenever we transform Cas9 to the plant, that become a transgenic. Then how these genome editing plants are the transgene free, right? So when we make a transgenic in a plant with this Cas9, that will with if we use whether gene gun or whether agrobacterium mediated transformation, that construct will get inserted randomly in any chromosome. For example, in rice, there are 12 different chromosomes, and if we made a agrobacterium mediated transformation and grow 5 independent rice transgenics, in all 5, the gene will be, the Cas9 will be inserted in any different chromosome. Maybe in first one on chromosome 2, second one in chromosome 11, or maybe on chromosome 5. Right? But the target gene is a fix. Suppose I am targeting aquaporin 1 in a rice. That gene is present on chromosome 6. Whether my Cas9 inserted into chromosome 2 or 11, that doesn't matter. That Cas9 will target on aquaporin present on chromosome 6 only. So whenever there is a change in on chromosome 6, we can easily segregate out that gene in a subsequent generation. For example, whenever we make a transgenic, that transgene is present in hemizygote condition. That means rice is a diploid crop that has a 2 set of chromosomes, and our transgene is present only in one chromosomal set. That becomes like heterozygous type of condition. And whenever we sell that, that will give us Mendelian segregation like 1:2:1 ratio. So in the next generation, automatically we will get a plant which has a desirable mutation, but the Cas9 and guide RNA is completely removed, segregated out. Then that become a transgene-free genome edited plant. In the process, first we develop transgenic, which has a Cas9 and guide RNA, but in a subsequent generation, we will have only the targeted gene modified and the Cas9 will be removed, and then it becomes transgene free. And once it becomes transgene free, now our government considers that as a like a normal variety. We can easily grow that outside in the field, or we can release that as a improved variety. But for transgenic, even if we wanted to do experiment in the field, it is not easy. We need to take a permission from government, and then we can at experimental level also we can grow the transgenic in the outside. That is also very difficult to get. That is the one thing how it is like a transgenic. When we compare genetic engineering approaches and conventional breeding has several limitations. Several limitations also several positive things also, because now after 3 decades of work on transgenic, we have only one transgenic plant, that is BT cotton, right? But we have thousands of variety in every crop. We have several improved variety. Those all varieties are developed with the conventional breeding. So we cannot say that the conventional breeding don't have potential. The most of 100% varieties we have from the conventional breeding approaches. When I am calling that conventional, that include the marker assisted or genomic assisted breeding. So breeding all the varieties which we have, that is from the breeding, right? But with genetic engineering, we can incorporate a new trait, which is not possible. The same three genes which I have shown in tomato, that we can combine in one variety with conventional breeding also. But that will take 7 to 8 years, one thing. And second thing, with the every breeding, for every single introgression of gene, that will bring in a unwanted genetic linkage drive. That means the genetic background of high yielding variety will change. That will not completely as it was earlier. So with the breeding, when we wanted to combine 3, 4 different genes, it will take like 1 decade of time, as well as it will change the genetic background of the high yielding variety. But if we adopt the multi-target genome editing, we can easily target 3 different genes, and that will not change any genetic background. There will be no genetic linkage drag. That is the main difference between this genetic engineering approach and conventional approach.
Anything else? Okay, so thank you very much, Professor Rupesh.
Thank you, sir.
We are delighted that you have shared your practical experiences on this biofortification, and he has nicely explained that the experimental work and the present work, future work, and even past work also, and the students are the testimonial of the same, that they did fantastic work. So, sir, we congratulate on
Thank you
that
very much, sir.
aspect, and again, we'll thank you, give thanks a lot for your time, and we give thanks to Madam Humira also, so to sparing the time, and we request you to you, sir, if the time permits, in
Yes.
the afternoon tomorrow. Today we have some packed schedule. So tomorrow afternoon we can have a short visit to sir's lab, and we'll try to show some experimental work, and you will see some reality of the uh, plant growth, and you will definitely enjoy that visit also. So sir, we can plan that also,
Sure,
if
sure, sir. Thank you.
time permits. So thank you very much again. Thanks a lot.
Thank you all. Thank you.